Hello, I'm Peter Karp, and welcome to the webinar on creating new Pathway Genome Databases. Here's an outline of this webinar series. In Part 1A, we'll describe what you need to build a Pathway Genome Database, such as what inputs are required, what outputs are produced, what software do you need, what can you do with a Pathway Genome Database once it's been constructed, and what are the inferences that occur during construction of a PGDB. Part 1B will show you how to use the software to do this database creation, and Part 2 will discuss how to refine and edit a PGDB to improve its quality. But first, real quick, let's talk about what is the difference between BioCyc and the Pathway Tool software. Well, BioCyc is a database collection. It consists of 20,000 total databases, of which 69 have been curated, their contents curated from 130,000 publications. And the biocyc.org website contains all of those databases and supports tasks such as information seeking, omics data analysis, and comparative analysis. Now, the Pathway Tool software was used to create the databases within the BioCyc website, and it actually powers the BioCyc website. You can install the Pathway Tool software locally to install a private set of the BioCyc databases, and to create Pathway Genome Databases from either your own genome data or from public genome data. Pathway Tools is an enterprise Pathway Genome data management system. It lets you update, edit PGDBs interactively to add new gene functions and pathways, for example. It lets you build quantitative metabolic flux models using flux balance analysis, and it really offers the same use cases as BioCyc, but Pathway Tools, in addition to being able to power a website, it can also run as a desktop application. Now here's the big picture of how Pathway Tools operates. It takes as its input an annotated genome, and it's the pathologic component of Pathway Tools, the inference component, builds a new Pathway Genome Database containing information from the annotated genome and containing new inferred information. Once we've created a new Pathway Genome Database, the navigator component of Pathway Tools can be used to search and browse and analyze this information in various ways. The Pathway Genome editors can be used to modify a Pathway Genome Database interactively, and the Metaflux component of the software can be used to create a quantitative metabolic flux model from a PGDB. The inputs to Pathologic to the PGDB building process consists of a genome in one of several alternative formats. We support multiple genome file formats. We recommend that you use the GFF3 format because one GFF3 file can capture a genome containing multiple replicons all in one file, and it handles complex splicing properly. Whereas the GBFF format, which is an extension of the earlier GBK GenBank format, although one file can capture multiple replicons, it sometimes does not handle complex splicing properly. The older GBK format, you can only put one replicon in a given file, so if you have more than one replicon, you have to supply a batch of files, which can be done, but it's more complicated. Similarly, the pathologic format that we invented also only supports one file per replicon. These files can be generated from multiple different genome annotation systems, including PROCA, RAST, KBase, and the NCBI prokaryotic annotation pipeline. In our experience, all of these annotation systems work with Pathway Tools. Another input is our MetaCyc database, which we'll describe in just a few moments. It's a universal pathway database, and it's usually built into Pathway Tools, so you don't have to do anything special to get MetaCyc. Now, how do we actually download a genome file from NCBI? Let's go through this real quick. We'll start at the NCBI taxonomy homepage, and we're going to search for the organism Acinetobacter baumannii and that particular strain. Then we're going to click, once we found it, we're going to click on Assembly on the right. We're going to click on the identifier of the assembly that we want. We'll take this one. We're going to go to the FTP directory for the refseq assembly, and then we're going to download the GFF file. The GFF file is right here, so we can click on that file to download the GFF file. And then the GZ means the file is compressed, so we need to uncompress or unarchive the file. Now, a little more detail about our MetaCyc database. It's the most extensive and highly curated metabolic database out there. It contains experimentally determined metabolic pathways, reactions, enzymes, and metabolites from all domains of life. We use it as a reference pathway database for the metabolic reconstruction algorithms in Pathway tools. It's a literature-based database with extensive references and commentary, and it's been curated from 75,000 publications. Comparing MetaCyc to KEGG, MetaCyc is analogous to the KEGG reference database. Both provide the universe of pathways and reactions for prediction in individual organisms from their genomes. But as you can see, MetaCyc has much larger content, has about 7 times as many pathways as KEGG does, about 40% more reactions, very similar numbers of metabolites, and the curation of MetaCyc is much more advanced, consisting of over 10,000 textbook page equivalents of mini reviews, summaries of pathways, enzymes, and so forth. Now, what are the outputs of the Pathologic software? It creates a new Pathway Genome Database that contains the input genome information, namely the sequence, the genes, the proteins predicted as part of the genome annotation, and it contains new inferred objects, namely pathways, reactions, metabolites, operons, and protein complexes, and evidence codes are attached to these new inferred objects to ensure that it's clear that these were computationally inferred. And I want to emphasize that the input genome needs to be annotated in the sense that it needs to have the positions of genes identified and the functions assigned for as many of those genes as possible, because Pathway Tools does not do genome annotation. Now, when you're creating your new PGDB, you have a choice as to whether to create it as a file PGDB or to store it within MySQL. The advantage of storing it in a file is that the setup is much easier. Okay, you don't need to install a MySQL server, for example, and configure it. But if you use the MySQL approach, you can have multiple users concurrently updating the PGDB. Different people can be connected to the PGDB and editing it at the same time. Also, the MySQL PGDB will capture in the database itself all update transactions. So you have a record of all the edits that were made over time and who made them. Another output of Pathologic is a whole set of report files that summarize the various inferences that Pathologic has made. What can you do with a Pathway Genome Database once it's been created? Well, the inferences that Pathologic makes usually provide new insights into your genome, such as what metabolic pathways are present. It provides a large set of search capabilities to help you find information you're looking for in the genome. You can browse and visualize the genome information using many different tools, such as tools that visualize the entire metabolic network and a genome browser. You can also compare genomes, compare their proteins, their metabolism, their transport capabilities. There's a whole series of omics data analysis tools. There's a metabolic root search tool, and you can build a metabolic model with Pathway tools from the PGDB. Now here's a broader description of the capabilities of the software. We divide them into these different categories: genome informatics, pathway, regulatory informatics, microbiome informatics. You can actually create a whole series of PGDBs for all the microbes or other organisms in a community. You can perform comparative analyses, omics data analyses, and perform a number of different data manipulation operations. Let's spend a few minutes just going into some of these in more detail. I'm going to let you read through the majority of these, but for example, under genome informatics, we have search tools for searching for genes, proteins, and RNAs, and there's a gene information page that summarizes all the information about a gene and its product. There are two genome browsers, a circular and a linear browser. Blast searches are supported, as are sequence pattern searches, and a multiple sequence alignment tool, and there's a sequence retrieval tool. Similarly, under pathway informatics, we have search tools for reactions, metabolites, and pathways, and information pages for all three of those entities. Pathologic will infer a metabolic network for an organism from its genome. It will also create zoomable metabolic map diagrams for each organism it processes. There's an interactive metabolic network explorer tool. There's a tool for performing metabolic root search across both single organisms and organism communities. There's a tool for pathway-based inference of the nutrient requirements of an organism. And you can create a metabolic model using flux balance analysis using tools such as both FBA and DFBA. Dynamic FBA are supported, as is flux variability analysis. There's a gap filling tool and a tool for assessing blocked metabolites. Okay, continue to read on if you wish. I'm going to go ahead here. Another thing you need, of course, is Pathway Tools itself. You need to install Pathway Tools locally on one of your computers. It is free to academics for research purposes, and there's a fee for commercial use. It runs on Linux, on the Mac, and on Windows. We typically release new versions of Pathway Tools twice a year, and we make source code available if you request it. Homepage is here. We have the following overview publication at arXiv, which describes all aspects of the software in detail. That's the place to look, although there are more focused individual publications. There's a user guide available in the software distribution, and you can fill out a license to obtain download instructions for Pathway Tools. And let me show you quickly how to navigate the licensing procedure. We start here at the download page. We'll go to the bottom and we want the software and data file license. So we're going to click here. And if we're coming from an academic institution, we're going to click here to obtain the license. We can first check whether our organization already has a license, in which case we can probably use the software under that existing license. In that case, just select your organization from this list and proceed with the rest. Otherwise, fill out the form and request a new license. So you can either request use under an existing license with this button or request use under a new license. Now, what are the actual inferences that Pathologic performs when it creates a new pathway genome database? We call this the extended paradigm of genome annotation supported by Pathologic. What people think of as the traditional paradigm of genome annotation is these first three steps: assembling the genome into large contigs from individual reads, running gene finders, and predicting gene functions. And it's the output of this process that is the input to Pathologic. Pathologic then performs the following inferences. It predicts the metabolic reactions of the organism, predicts metabolic pathways of the organism from the reactions. Now, when pathways are predicted, there are typically some steps in the pathway for which no enzyme has been identified in the genome because of the incompleteness of genome annotation. Pathologic will try to predict which genes in the genome fill those pathway holes. It will also predict the transport reactions of the organism. It will generate a metabolic model for the organism from the inferred reaction network. It will predict operons of the organism, and it will predict protein complexes of the organism.